NIH in
My
the
name is
US of A.
Justin Choi, and we are pleased to present our research topic of a framework to discover the genomic risks of Alzheimer's disease. Alzheimer's disease, also known as AD, is a type of dementia which results in a loss of cognitive function and affects 47.8 million people in the world. Typically, AD is diagnosed using MRI and CDR, clinical dementia rating. However, genetic biomarkers from organs such as blood, hippocampus, and peripheral tissue are more vital because typical methods have limitations for diagnosis. Thus, we introduce a pipeline to discover the unique gene expressing patterns in AD patients. Using RNA sequence data, we trained support vector machines, known as SVM, and neural networks, and evaluate their performances to detect unique expressing genes in AD patients.
For the experiment, an RNA total expression dataset of middle temporal gyrus samples from 79 AD and non-AD patients was used. Additionally, the experiment was implemented by R language. After calculating the fold change of selected candidate genes in the RNA sequencing dataset, genes with top highest fold changes were selected as features for machine learning algorithms. We repeatedly ran an SVM and a neural network in increments of 1,000 with top gene features each time until we got the highest accuracy. Then, using the top ranking candidate genes, we validated their genomic functions through a gene ontology analysis.
After running these repetitions of both the SVM and neural network models, we observed that both models displayed a high accuracy in classifying AD and non-AD patients, with the SVM yielding an accuracy of 0.855 and the neural network yielding an accuracy of 0.950. These high accuracies suggest that the levels of gene expression have a possible important role in AD diagnosis by using a gene which is expressed differently in AD and non-AD patients. Accurate diagnosis is possible. In addition to the SVM and neural network, from the gene ontology analysis, also known as the GO analysis, we observed that the top-ranking candidate genes were all related to the functions of synapses in neurons, showing that a malfunction in synapse function could be a possible risk leading to Alzheimer's. For future research, we would like to work on increasing the accuracy of diagnosis with gene expression levels through creating and using deep learning models. In addition, we would also wish to expand and test genes present in other areas of the brain. Thank you for listening to our presentation.
Thank you, Grace. Is there any question? Yes. Okay, we have a first question from Ayush. Could you please tell us how was the data split between Alzheimer's disease and non-Alzheimer's disease classes?
So in order to split the data, we used SVM, which classifies data using hyperplane in a data dimension, and also using neural network. But in order to train these, we used the expressing features of the selected genes as features, and we repeated training increments of 1,000 with top gene features each time until we got the highest accuracy in classification and splitting between the AD and non-AD.
Okay, maybe just another one question. And the last one, because we won't have much time. Could you tell us a bit more about the neural net architecture used?
So our neural network was kind of like the basic neural network that usually is used. We just classify data through the changing weights among nodes and using active function of each node in layers. Of course, we just trained it like the SVM before, just using the expressing values of selected genes and just increment it to 1,000 to get the highest accuracy possible. We did hit like a point where adding more genes did not increase the accuracy at all, which was like the saturation point of the neural network. But otherwise, yeah, that was how the neural network worked out to be.